The overall goal of this procedure is to utilize a natural vector, the deer tick, for experimental transmission and xenodiagnosis of the Lyme disease agent Borrelia burgdorferi. This is accomplished by first producing ticks that harbor the bacteria. Once inoculated, the ticks are applied to the shaved area of a mouse and then housed in specialized caging. Fed ticks are then retrieved from the cage water. For non-human primates, a specialized containment device is constructed and applied before the ticks are placed on the skin. After 5 days, the device is opened and fed ticks are collected. Ultimately, samples can be collected and analyzed by various methods to confirm infection after tick feeding. Tissues can also be cultured, sectioned, and stained for immunofluorescence.
Visual demonstration of these methods is critical, as the multiple steps involved are actually quite different between the animal models used. Demonstrating the procedure will be myself, Dr. Britton, Grasperge, and Mary Jacobs.
White lab coats with elastic sleeves, gloves, and disposable bouffant caps should be worn when working with ticks. To begin, retrieve nymphal ticks that have been stored at 23 degrees Celsius for 4 to 6 weeks post-larval molt. Place double-sided tape on the bottom of a small petri dish and secure the ticks ventral side up on the tape. For inoculation, B. burgdorferi are grown to mid-log phase in BSKH medium containing 6% rabbit serum. Glass capillary tubes are prepared using a tube standardized to the tick mouthpart as a sizing guide. When ready, mix the B. burgdorferi culture and insert the tip of the capillary.